fundamentally, everyone in the company lives or dies by the success of the company. And so this kind of breeds a sense of collaboration and teamwork that I think we've tried to replicate in academia, but it's inherently much harder. To an extent, I feel like I still feel a little bit like an academic in well, probably a sheep in wolf's clothing.
Welcome to the Nucleate Podcast. My name is Sam Kessel, and on this episode, my co-host Anastasia Yanas and I speak with Dr. Peter Campbell, the CSO of Quotient Therapeutics, a flagship pioneering company. He's the academic co-founder who recently made the jump full-time to Quotient from the Wellcome Sanger Institute, where he was the head of cancer, aging, and somatic mutation. Dr. Campbell has received numerous accolades over the course of his career, including his election to the U.S. National Academy of Medicine in 2022.
Thank you for joining the Nucleate Podcast, Peter.
Thank you. Great to be here and looking forward to the conversation.
Now it's a little bit of tradition to start with the background. So, kind of going backwards to where you grew up and how your passion for science started.
Yeah, so I grew up in New Zealand, in a university town. My schooling was, I think, fairly traditional for New Zealand schooling, a mix of arts and sciences all the way through. And I didn't really know where I was going to wind up, except that I was always better at sciences than at arts. Within sciences, I was pretty open-minded as to where I might go. And I applied to medical school and was accepted to medical school. And so kind of without really much thought, wound up day one at medical school. And frankly, I hated it. It was rather intellectually unsatisfying. There were bits of the kind of pre-medical training that seemed irrelevant to being a doctor. And so I begged my lecturers to allow me to stitch in some other courses and wound up a dual major in medicine and maths. And I kind of always wanted to do research, but actually medical school got better, and as time went by, and I enjoyed interacting with patients, and I enjoyed the clinical aspects of medicine. I sought advice as I was kind of coming out of med school. I always wanted to do research, but the advice that I got in New Zealand at the time, it's probably would be different advice now, but at the time the advice was finish your specialist training, choose what you want to be, what kind of specialist you want to be, do that clinical training, and then do your research as you're coming to the end of that specialist training. So I duly did that. I trained in hematology in Australia and New Zealand, and then as I was coming to the end of that, started to think properly about doing some research. And at the time, the person who became my PhD supervisor, Tony Green at the University of Cambridge, had emailed out a few of his contacts in Australia and New Zealand saying, if you have any young hematologists coming through wanting to do a PhD, send them my way. And so my clinical boss at the time in Christchurch put me in touch with Tony, and so I came to Cambridge to do a PhD with Tony, and it was fairly traditional. I did a mix of bench work and some data analysis work. And I guess I realized doing this that I was just a disaster at bench work. I did not have the patience. I did not have the attention to detail. I did not have the fine motor skills. Nothing about bench work was made for me. And this was a bit discouraging. But alongside that kind of realization that was not the life for me, I also realized that, building on the kind of maths and stats background that I'd had in my undergrad life, combined with the medicine, it was a fairly natural jump, I guess, to go from that kind of PhD to what would now be called a bioinformatics postdoc. The time bioinformatics barely existed. This idea that you might specialize in computational analysis of data was not something that anyone would really imagine a career could be. We had, during my PhD, we'd worked with the Sanger Institute to be kind of doing systematic sequencing of exons using capillary PCR and capillary sequencing. so fairly old-fashioned by today's standards, sequencing technologies, we sequenced a bunch of kinase genes in the particular cancer that I was working on, and had discovered mutations that kind of unlocked that disease. So it was a sort of fairly short leap from the University of Cambridge to Sanger Institute for my postdoc, where I was kind of focused on genomics, and this really was my calling, I guess. This is it is very data rich. It's unbiased. Doesn't a lot of it can kind of happen in a factory, if you like, and so doesn't need particularly impressive wet lab skills. And so it seemed like a pretty amazing opportunity for me. This amazing opportunity, as I was considering it, was enhanced when I joined at Sanger, because literally in the first week at Sanger, my boss said, there's this next series of sequencing platforms coming, and we've just got access to the first one of these. And this was what was called Solexa in the day, but later was bought out by the startup that was bought out by Illumina, that became the kind of workhorse of genomics for the next 10, 20 years. I was lucky enough to do one of the very first runs on this platform at the Sanger Institute, and the first time that anyone in the world had sequenced a cancer using this new technology. And so suddenly we had this incredible data set from the first cancer cell line that we sequenced, with massive data that there were no tools to analyze. So we set about, basically, this was my postdoc, was writing tools to analyze this data that no one had ever seen, didn't know what the kind of patterns of damage or artifacts or anything were, and try and extract some sense from this, piles and piles of data. And it was extraordinary because, the data was so clean and so accurate that we could suddenly see things in these cancer genomes that you couldn't possibly have dreamed of seeing a year before. I mean, it's just incredible to be able to sequence that whole spectrum of mutation that one sees in a cancer, and see it kind of come out of the mist of this morass of data. So I was hooked, frankly. This was my calling. This was what I wanted to do, and it was a fairly natural thing as time progressed, and I guess my career progressed to kind of stay in that space. And it was an incredible time to be doing research, because we were kind of right in on the ground floor of this revolution in genomics. We were seeing things that you couldn't possibly have imagined being able to see within a lifetime. Suddenly, the kind of mists of confusion around how cancers evolved and how they developed was parting, and we could see the kind of broad outlines of what drove cancer. And so I guess from about 2008 to 2015, we really kind of systematically explored that landscape. Because we weren't the only people doing this. Eventually we published the first genomes in the first cancer genomes, and kind of led the way in a sense. But the rest of the world kind of joined in through the Cancer Genome Atlas and the International Cancer Genome Consortium. And there was a massive worldwide systematic effort to catalog the genetic changes that drive all major adult and childhood cancers. And it was an incredibly exciting time to be part of that effort. And admittedly a small cog in that effort, but to feel that there was a kind of that this was going to rewrite the textbook of cancer forevermore, and to be part of that was pretty amazing. So that was my sort of academic journey up until about 2014-15. After that, we could kind of see the end of this discovery phase of cancer genomics coming. We were not sure where it was going to go. We knew that most of the major cancer genes had been discovered. The kind of major mutational processes in cancer had been catalogued. The patterns of the way that cancer evolves had been documented. So we were sort of seeing the end of that discovery phase of cancer genomics coming. It was clear that the next phase of this was moving the cancer genome sequencing from the university into the clinic. We were, I guess, not sure about what an academic's role in that would be. We actually wound up founding a company to try and do this, which was called 14M Genomics. It sadly fell by the wayside after 2 or 3 years. But that was my first brush with entrepreneurship, if you like. And it was, and I think in a sense, much of the way that cancer genomics revolution that happened in academia has moved into the clinic has been through a kind of commercialization route. There are some academic kind of health system collaborations doing this, but mostly it's happened through a commercial lens. And I think that is we can discuss the reasons why that is so, but I think that was how we felt it would have to be. But that left the kind of academic program wondering, well, what comes next? And, we had documented the kind of buildup of mutations in cancers as you go through life. And we'd begun to wonder, well, it's obviously the case that the cancer has mutations. What on earth is happening in all of the other cells in your body that aren't cancer cells? And we began to develop technologies to look at mutations in normal tissues. There were various technical developments that were required to do that, both on the wet lab side and on the dry lab side, which we were pretty well placed to deliver on. And so we pivoted really to trying to develop these technologies. And then once we had built those technologies, the same excitement came back again. Suddenly, all of the things that we had been just gobsmacked at that we were seeing in cancer genomes, we were seeing writ large on every cell in your body. So, we saw the kind of buildup of mutations in every cell, even post-mitotic cells in your body as you go through life. And as we systematically documented these changes in every organ system, each organ has its own story to tell. We could see in skin, we could see the damage from ultraviolet light. In lung, we could see the damage from tobacco smoke and what it does to the genome in every lung cell. We could see in blood, we could kind of see the way that the mutations were changing the dynamics of blood development and so on. And this landscape, again, just emerged out of the mist with the right set of technologies and tools. And as we kind of documented this in normal tissues, we began to wonder, well, what happens in aging? What happens in diseases other than cancer? And again, there's a whole new landscape of stuff to be found. That really is the story from 2015 of my academic career, which kind of led to this sense that the genetic changes that are occurring in all of our cells throughout life are creating this enormous genetic diversity in our organ systems. And then this shapes the way that cells respond to whatever's around them. So if there's a disease, if there's excess calories, if there are DNA damaging agents, if there are infections, if the patient's taking various drugs, the cells kind of use the mutations, or the mutations drive the cells to respond differently to these different environments. And we can now read this out at incredible scale. As we were kind of uncovering this treasure trove of information, we began to wonder, does this have any clinical relevance? Is this useful information to know? And at the same time we were wondering this, Flagship Pioneering reached out and said they'd noticed some of our papers and they'd been wondering this same thing. And it was a fascinating conversation because we were wondering this with a set of data that hadn't yet been published. They were wondering it from data that we had 2 or 3 years ago. So we were super impressed that they had picked up on this opportunity from data that was 2 or 3 years out of date, and we'd only noticed this opportunity when we'd really systematically gone after some diseases. And so it felt like a very interesting marriage of minds, in a sense, that they brought this kind of intellectual commercial vigor, and we brought a series of tools and techniques and algorithms, and we could bring them together to do something. And that's really the sort of genesis of Quotient Therapeutics. Anyway, that's a long way from growing up in New Zealand.
So this was an amazing overview from basically where you grew up and up until now. So I think my questions, like you answered almost all of them, but let's go back like to specific things that you said. So first of all, hating medicine, especially in the first years, I think a lot of our audiences who are right now in the medical school, they can they feel the same way, because in the beginning you just learning sciences, learning things that you don't even understand why they're relevant. So for audiences who are right now struggling with the first years of medical school, it will be better. So what you said is basically that you were always very curious and very open to science. And was the move into genetics like it felt a little bit like you said this was your calling and that the opportunities presented themselves, but was it something at the time that you were pursuing yourself as well, or was it more that you were just grabbing the opportunities as they came and then you found out that this is something that you're really excited about?
Yes, I think probably the latter. That to an extent it was opportunistic. But it was kind of but it was fairly instinctive, I guess. So what really called me about genomics, and this had kind of figured largely in my PhD, was the fact that with genomics you can, one, be unbiased. So you don't have to make some hypothesis or kind of make some prediction and then test it with all of the biases that are inherent in that process, and that I think have in some senses, in some fields, slowed down science. So you can be completely unbiased and agnostic. You can just show me where the money is. Basically, that's one advantage of genomics. The second one, which is often not discussed, but I think is absolutely critical, is that genomics is fundamentally causal, right? So the everything stems from your genome. You know, it's like there's so many pieces of evidence for this, but you know, essentially the naked DNA sequence, that the code of letters, is enough to encode the entire individual and the way that every cell behaves within that. And that, when you're kind of doing basic science, is a really exciting thing. And every time that I've dabbled with stuff other than genomics, like if I go and do some transcriptomics or some mouse model, heaven forbid, I just haven't done many of these things in my academic career, but every time I have, it is much harder to anchor that the things that you see in a transcriptome or in a cell model or whatever in a causal framework. But genomics gives you that causality, and that is fundamentally important for so many of the kind of basic insights into the way that our cells behave. And the third thing that's really exciting about genomics, from someone who's got a sort of maths bent, is that it's digital. So, it's a code, and that code is enormously complex, but it is decipherable. And we've, I think, scratched the surface of deciphering that code. We know the genetic code, as in which bits code proteins and how that decoding works. We're making kind of inroads into gene regulation, but the kind of wider network properties of it, I don't think we're close to decoding yet. And so there's a kind of another generation or two of work to do that decoding. So those three properties, the kind of the unbiased nature, the causality, and the kind of digital nature of genomics is what, for me, kind of drove that kind of instinctive calling.
That's amazing. And let's build a little bit of foundation for our audience to understand basically what you're doing. What you said, so basically that you found out that like in cancer, you have a lot of mutations, and then you also found out that in our normal cells, we also have mutations. So let's depict it a little bit, because the normal way we know, okay, there's a mutation, this causes cancer or something like a malignancy. But when you say the normal cell accumulate mutations as well, so what is the deciding factor between this is something that benefits the cell or versus this is something that actually causes harm and ends up being a cancer that might kill you?
Well, like so many things in biology, this all comes down to evolution. And, we well understand the process of species evolution, and it has become clear that somatic cells experience the same processes. If you think about what are the requirements for evolution, there are three. There's basically you need competition for scarce resources. So competition between individuals, and in a somatic cell setting, those individuals are the cells themselves. So the cells need to be competing for scarce resources. There needs to be some form of heritable variation within that population of individuals. So in this case, it's somatic mutations. So the genetic changes that our cells acquire as they go through life generates heritable variation within the population. And that heritable variation needs to lead to phenotypic differences. So the mutations need to change the phenotype of the cells. If you have those three ingredients, evolution will happen. That's a fundamental property of those three tenets. And what we've demonstrated is that in normal cells, all three of those apply. So you can see there's competition for scarce resources because cells are under enormous selective constraints. They can't grow out of their little layers or their little niche, whatever. There's a whole heap of things that constrain their growth, things like telomere attrition or kind of spatial density dependent crowding constraints, et cetera, et cetera. There are all these constraints on how cells grow. We showed that all cells accumulate mutations as they go through life, and at a reasonable rate. So, your average blood cell accumulates about 17 blood stem cell accumulates about 17 mutations per year per life. Even your neurons are accumulating mutations at the same rate, even though they're not dividing, but they're still accumulating mutations as you go through life. And that variation leads to phenotypic effects. We can measure that with transcriptomes. We can measure it based on other properties of the cells. So evolution happens, and basically what we observe when we look at all different organs is that there is this fight going on between individual clones within the cells, within the organ system. Some clones are better suited because of a genetic change to living in a particular environment. And this means that over time, and in particular as we age, you get these shifts in clone frequencies. Some clones come to dominate, some just wither away. And this is all driven by, not all driven, but much of it is driven by selection, Darwinian selection, acting on the genetic changes that they've acquired. Probably also epigenetic changes, particularly in the form of DNA methylation. So in those three tenets of Darwinian evolution, I talked about somatic mutations as a heritable variation, but actually DNA methylation is heritable, and it varies from cell to cell. So there's no reason why Darwinian evolution wouldn't also act on DNA methylation changes. So this kind of shift in kind of clonal mix within an organ system is essentially completely responsive to the environment that organ lives in. And one of the kind of amazing things that we've kind of been able to document as we've kind of expanded our studies is just how plastic that environment, that relationship between the clonal architecture of an organ and the environment is. So, for example, if you give some form of drug like a chemotherapy or immunotherapy or many other medications, you change the selective landscape within the organ, and suddenly you see all these clones emerge with particular mutations in particular genes that you don't see in healthy people or people not taking that drug. If you look at the liver of someone who has obesity, and where their liver cells are being exposed to all of this fat lipid toxicity, then you get a whole set of mutations that protect the liver cells against that fat toxicity. If you look at someone born with a germline defect, say a ribosome defect, that would normally lead to bone marrow failure, and you look at the blood cells that survive in the most severe children with these defects, every single stem cell that survived has found some mutation that enables them to revert the ribosome defect. So there's this extraordinary kind of relationship between the pressures that cells experience, this massive genetic diversity that our organs have, and the kind of how cells then can adapt to those different environments. And just it's amazing how plastic this is. If you take that drug away, if you stop smoking, the clonal landscape of your lungs will revert back to how they were to an extent.
That's very amazing. And so right now the question is, you have so many mutations per cell that you said, like amazing numbers, and you like also from the computational side. So you right now have all these mutations and you have this code that you have to shipper, and then you are presented with a spreadsheet or kind of a list of, as I said, all these mutations. So how do you know what is an important one, what is something that is just there but doesn't have any effect? So can you tell us a little bit more on that?
Yeah, that's a really good question. And let me just give you some numbers just to kind of contextualize this genetic diversity that we see. So if you take T cells, T lymphocytes, you have about 100 billion or something T cell clonotypes within your body within an adult. By the age of 60, the average mutation burden of those T cells will be 2,000 mutations. Your genome is 3 billion bases big. So what that means is that every single base in your genome will be mutated something like 6,000 times within that population of T cells. So every single mutation that you can possibly imagine will be present in your T cells. And that is more genetic diversity in one subcompartment of one organ of one individual than has ever existed in the germline of all humans that have ever existed. There's more genetic diversity within one subcompartment, one organ. And when we sequence T cells in an individual, let's say they're T cells of an individual who has cancer, or T cells of an individual who has an autoimmune disease, or T cells of an aged individual, what we're looking at is are there particular genes where we're seeing more mutations than we would expect by chance? So if you can imagine, you take those 100 billion clonotypes, you sprinkle mutations in their genome just through the natural kind of mutational processes, and that most of those are going to do absolutely nothing to the T cell, but there'll be one little sprinkle here and another little sprinkle here that will say to that T cell, expand, proliferate, go to this inflamed tissue, go fight this infection. And then that mutation, that cell will expand, and we will see more copies of that mutation and mutations in that gene than we would see in the others, because those cells have expanded. And so essentially, I mean, there's a whole heap of statistics that underpins this, but that is basically the principle. We are looking for more sprinkles in specific genes than we would expect by chance.
Okay, it's a very understandable explanation. Thank you for that. So for our entrepreneurs in the audience, amazing science, amazing opportunities, amazing, like a lot of interesting facts. So what is the most exciting use case that you see for everything that you find out?
Good question. And this is something that we're wrestling with at Quotient. We were writing lots of papers on this landscape that we were uncovering, and lots of grants to kind of follow up on individual findings. And the kind of last paragraph of each of those papers or grants was, oh, and this might have translational relevance. And we weren't doing that translation. We were paying lip service to it. We were kind of pointing the way, if you like, but we weren't actually doing it. The fact is that much of this selection that we're seeing is telling you a huge amount of causal biology about the way that cells are responding to different environments. And a major environment that cells are exposed to is obviously disease. And so we believe that you can learn a lot about the biology of a disease from seeing how cells respond to that disease process. So my favorite example on this is in the liver disease, because that's where we kind of started with this. So if you take alcohol-related liver disease or obesity, metabolic syndrome-associated liver disease, in both of these, what you tend to see is that the liver accumulates fat. So the alcohol in the alcohol-related liver disease, the alcohol gets converted into fat. There are various other pathways, but basically the kind of final. And then in metabolic syndrome or obesity-associated fatty liver disease, the calories are coming in from the diet and they're being processed in the liver into storage fat. And that fat buildup in the liver is enormously toxic, something called lipotoxicity. So what we found, and this was started in the academic program, but we've pursued in the company, is that this kind of process of fat buildup in the liver, this disease, if you like, in the liver that's causing toxicity to the hepatocytes, represents a very strong selective pressure. So suddenly hepatocytes are dying left, right, and center because they've got too much fat and they've got all this toxicity as a result of that. And liver cells have this enormous genetic diversity. In fact, they're quite highly mutated liver cells. And so there might be cells that kind of protect, there might be mutations that protect the liver cells, essentially tell the cells not to make this fat or not to take the fat out of the circulation, out of the diet. And those cells, they won't suffer the toxicity that all of their neighbors are suffering, and then those cells will kind of survive better than the other ones. And so the mutations that we see in this setting of fatty liver disease are a whole bunch of essentially mutations that tell the cells not to process fat in different ways. And so you've gone from you can see that these mutations essentially protect the hepatocytes, and therefore you can imagine, if you were going to think about this therapeutically, you can imagine that you could kind of mimic the effects of these mutations for all hepatocytes, and then maybe be able to spare them that damage and that toxicity from excessive fat accumulation. So that's the kind of bet that underpins Quotient, that by sequencing cells at the coalface of the disease, the ones that are right there facing the disease process, you can find mutations that tell you fundamental properties of that disease process, and then that will give you clues as to how you might modulate or block that disease process. And there are lots of different ways that mutations can do that, and there are lots of different ways that we have to think about, interpreting the mutations that we find. But fundamentally, that's the bet, that you have you're making causal insights into the pressures that cells at the coalface of disease are facing. And so that's I think, a huge opportunity. And actually, one of the kind of exciting things about it is that there are lots of diseases where this paradigm is likely to be quite informative, and so it really feels like a hugely exciting opportunity to explore all of these diseases.
Yes, agreed on that. So once you come closer to the clinical applications, would it be something that is on the level of personalized medicine? So you would need to sequence the genome of every patient, or is it something that you can scale and apply more broadly?
That's a really fascinating question, and it's one that we're really wrestling with, actually, in the company. And let me make this kind of concrete for you. So in that liver disease setting, where I was talking about, say, the alcohol-related liver disease and the obesity-associated liver disease, you get to the patients get to that fat accumulation in the liver through different routes. So one might get there by alcohol, and the alcohol then goes through some sort of de novo lipogenesis to get converted to lipids. Other people get to that fat buildup through eating, McDonald's and pizzas and all of these other fatty foods. And so essentially they're directly ingesting their fat, and then the liver cells are taking it out of the portal bloodstream and processing it. So that fat buildup is happening in different routes. And what we see when we sequence patients' livers with fatty liver disease is that you have this phenomenon of within patient convergence, but between patient divergence. So what we will see is that the same gene gets mutated again and again and again and again within a given patient. But then if we go and look at the next patient, they'll have a different gene that gets mutated again and again and again and again. And the reason for that is that we think is that, let's say, take your alcohol content where they're getting to their fat through de novo lipogenesis. The pathways that you see mutated in those patients are genes, for example, involved in de novo lipogenesis. So genes that are involved in converting alcohol to fat. On the other hand, in the setting of kind of where your dietary fat excess, what you tend to see are mutations involved in inhibiting insulin signaling, because what the liver cells are basically being instructed by insulin, take the fat out of the bloodstream and deal with it. And so cells that can basically switch off how they respond to insulin, they don't take the fat out of, they become insulin resistant. They say, I'm not taking the fat out of the circulation. They're perfectly fine. So you see this kind of difference between patients that is even more than just saying that they have fatty liver disease. It's saying how they get fatty liver disease, what the kind of pathway to their fatty liver disease is. And so coming back to your question, it's a pretty long-winded answer like all of them. Coming back to your question, what I think this will mean is that we can imagine a world in which you're doing personalized medicine like we were doing in oncology, where you would kind of sequence a cancer and find the specific genes that were mutated in that cancer. you can imagine doing the same thing in these other disease settings where you look at what are the properties of the disease the disease in this patient, and what's the best therapy going to be? What does this tell me about the right therapy to use for this patient? So I do think that there is a kind of personalized, a more personalized medicine aspect to this. There are lots of logistic challenges in doing that, like, biopsying people's livers and then sequencing them is a non-trivial undertaking. But there might be other ways to do this, and I think that we will probably wind up for a lot of these diseases actually kind of trying to marry the patient to the right therapy for them. And I hope that somatic genomics will play a role in that marriage process.
Okay, so right now a little bit provocative question. Based on what you said, and you also said that the cells adapt to our environment, and basically, if I would go out and eat McDonald's all day, then my cells would adapt to that. So question is, if I stop eating McDonald's, wouldn't we go back to normal?
We don't know. So what I would say is that the genetic changes are permanent. So once a cell has one of these mutations and it's expanded, that clone will always have that set of mutations. But what we have seen, so for example, we see this really strongly in lung. So if you take bronchial epithelial cells from smokers, what you find is that, basically every cell has, a few thousand mutations that and the majority of which have come from tobacco mutagenesis. If you take an ex-smoker, what you find is that there are those cells, they still exist, they've survived with the tobacco damage, and but there's a bunch of cells that have kind of emerged that have a much lower mutation burden. And if you look at the kind of the pattern of mutations in the cells in the smoker, you'll find lots of mutations that you would see in a lung cancer genome. So all of the sort of things that you would see in a squamous cell carcinoma of the lung, you can see in the normal bronchial epithelium of smokers. These cells with what we call driver mutations or cancer-causing mutations are much less frequent in the ex-smokers. So I think probably what happens is that the cells with these mutations kind of have their selective advantage only in the presence of tobacco smoke. And once you remove that tobacco smoke, that advantage disappears. In fact, it may even be a disadvantage then if there's no tobacco smoke. And then all of these other more healthy cells kind of repopulate the bronchial epithelium. And we think that is one of the reasons why if you stop smoking, your rates of lung cancer gradually over time get closer and closer to as if you'd never smoked, because essentially you get this kind of regeneration of more healthy bronchial epithelial cells with lower mutation burdens and the absence of these cancer-causing drivers. So yes, I think it's quite plastic. And so yeah, in your McDonald's example, it may well be that if you do stop eating that high calorie diet, then those cells with this selective advantage will no longer have that advantage, and they'll be outcompeted by healthier cells.
Do you think it's all just evolutionary, like the cells themselves die off due to selective pressures like you highlighted, or is there also a role for the immune system in killing off some of those unhealthy mutations once the external environmental piece is gone?
Yeah, it's a really good question. We don't know. I suspect, I mean, clearly the immune system is good at weeding out, or is good, but it is involved in weeding out cancers and cancer cells. At this earlier stage, my sense is that the immune system doesn't really care until clones get large enough that there's enough of an antigenic stimulation to respond to them. So I suspect we have a bit of evidence for this, but it's pretty weak. I suspect that by and large, in the early stages of this kind of Darwinian process, actually the immune system's pretty agnostic.
That is a really interesting dynamic. Now, when we're talking about Quotient and all these insights that you've highlighted, and I know that you guys don't have like a really disclosed pipeline at this point, so feel free to talk at a high level here. But when it comes to turning these insights around somatic mutations, and you've highlighted examples where there's positive selective pressure and negative selective pressure, when you're turning in these mutations on both positive and negative sides, how do you think about turning those into therapeutics?
Yeah, it's a fascinating question. This is a very live debate within the companies, you might imagine. So, the platform, if you like, is the sequencing platform. So we can essentially now take tissue. We love to work with tissue of diseased organs. So we like to work with the cells that are right at that coalface of the disease process. So we take those disease biopsies, and we now have an ever expanding array of ways to isolate cells with particular features or phenotypes, and we have ever increasing ways of sequencing them to identify the mutations that they have, and then ever more sophisticated computational platform to analyze that sequencing data and extract the genes where we're seeing too many sprinkled mutations, if you like. So that platform is now, it's not perfect and we're still continuing to enhance it, but it's humming pretty efficiently. And we're doing this across a lot of diseases. What we are finding as we do these diseases is that we are getting genes targets out of this that are, some of them, they're pretty well-known targets in the particular disease area, and every big pharma company has an asset in this on that particular target. And for us, it's nothing more than okay, well, our platform worked, but told us what we already knew. Then we find a whole bunch of genes where there's literally like 2 papers on PubMed, and we go, what on earth are these mutations doing? Where do we even start with this gene? And those we just don't know what to do with. And then there's this sort of sweet spot for us, which is where you have maybe, I don't know, 40 or 50 papers in PubMed, and maybe 5 of those have said something about relevant to the cell type that we think is involved in the disease, or has some sense that it might be relevant to the disease process. And then we kind of ask a few questions, basically. So we have this kind of quite detailed target assessment process. And the questions that we ask are really, do we think these mutations, what are these mutations doing to the gene? Are these loss of function mutations? They're just killing the gene? Are they somehow gain of function? They're just activating the gene? Or are they modifying function? Are they doing something really quite subtle to the way that protein functions? And that's obviously quite important. And the second question we ask is, are these mutations driving the disease process? In other words, they're causing the disease, or are they protecting the cells from the disease process? So the example that I've talked about in the chronic liver disease setting is really around is an example of mutations that protect the cells against that lipotoxicity. But we also believe that there's a set of mutations that can drive disease biology, not in liver disease so much, but in other diseases. And so those are the sort of 2 broad questions, because that determines how you think about a therapeutic strategy. So if you believe that your mutations are protective and that the mutations are loss of function, then what you want is to develop an inhibitor of that, because you want to mimic the effects of the mutations in all of the cells in that organ. If, on the other hand, you have a set of mutations that you think are gain of function and are driving the disease biology, then you want to block that. That gene, because you want to switch the activity of that gene off. And then remove the cause of the causal kind of drive of the disease. So that's the kind of broad outline. Now, it won't surprise anyone when I say that we look at the mutations. There's a kind of we dig out what we know about these genes from the literature, from germline studies, et cetera, et cetera. In the end, quite a lot of the the kind of conclusions that we reach are tentative from that process. There's only so far that navel gazing and in silico work can get you. In the end, you need to get your hands dirty and test those hypotheses in the lab. And so we're kind of ever expanding our ability to take to test the effects of these mutations in vitro and in vivo, to really understand and get conviction that these mutations are fundamentally important to the disease biology, and that modifying or somehow therapeutically responding to this is going to make a meaningful difference in the disease process.
So is it fair to say then you are exploring both options? You're exploring both inhibiting disease-driving mutations as well as augmenting protective/preventive measures.
Exactly.
It's just going to be disease specific. It's going to be based on how, to your point, some early in silico studies, but as well as what you're seeing in the lab setting and animal setting, and getting a sense of whichever, you're agnostic to which approach, but whichever one is driving the best result is the one you're going to go after. Is that fair to say?
Yeah, that's fair to say. Exactly.
Great. So when you're thinking about these kinds of mutations, have you at all thought about which modalities you intend on using to drug these targets? Or is it going to be, again, case by case basis, depending on the mutation, depending on, if it's hard to drug with a small molecule, use biologic, or if it's gene editing, you'll use gene editing? Just curious on how you're thinking about actually going after these targets.
We are completely modality agnostic. I think, and when we're pretty open-minded, we're optimistic, I guess, as a company that the kind of drug design, drug development, the kind of that side of things is becoming increasingly commoditized. We are not the commodity provider for that. But we believe that, and I think one of the kind of exciting things about the company is that if this is true, that drug development and drug design is becoming increasingly straightforward and with a kind of wider range of modalities, then having good quality targets actually is going to be much more rate limiting and much more kind of success determining. And that's one of the reasons that we've kind of positioned the company in the way that we have. So yeah, we're agnostic, I think, and we don't play into any particular secret source in this, or secret kind of ability in this space, where we would be using the same modalities and the same approaches that others would. We may well wind up doing a lot of this in partnership, either with other flagship companies or with big pharmaceutical companies or with CROs or whatever. And I think we'll be pretty pragmatic about the approach to that. Our secret sauce, if you like, is more in the kind of target ID space, target ID, target validation space, and then kind of the genetics gives you quite a lot of clues often as to where you might want to drug a protein. If you have a particular sort of domain or hotspot of mutations, that tells you a lot about where you want your drug to go. And we also get information because we know what cell types we're sequencing. We also know what cell type we want to sequence, cell type we want to drug, and certainly for some of the biologics, that opens up interesting possibilities for quite bespoke therapies. So we can use the genomics to inform on that kind of modality design, but we don't. I think we will be pretty pragmatic about which one works best in a given setting.
Yeah, that makes sense. So based on the target you're discovering, based on the cell types, based on, if you're going after the protein itself or RNA, there's different opportunities to drug the targets. And as you said, finding new targets is not so easy nowadays. So the fact that you have a platform that enables you to do that allows you to be very creative and pragmatic around how you drug those targets,
Exactly.
which I find really interesting. So this actually brings up an interesting question that has come up before in the podcast episode with Dino around how you think about being a platform kind of company where you're supporting, or in this case, doing target discovery or maybe some interesting initial steps, but then ultimately partnering with larger pharmas and biotechs, et cetera, like you mentioned, in the Flagship network to actually pursue those drug development. So curious on how at this stage you're thinking about whether you view yourselves more as a discovery platform that will eventually hand off or partner with another entity in order to develop a drug pursuing that target, or if you see yourselves developing your own drugs internally. It sounds like you're still on the internal side a little bit in terms of drug development, but curious on how you're thinking about that strategy.
Yeah, and it's a pretty existential question for us, as you might imagine. I jumped ship from academia to be CSO here at Quotient at the beginning of January. So I'm 4 months into it, and I would say I'm it has been amazing in those 4 months. I've talked to a lot of people, as you might imagine, in this world, and they say that they're pretty consistent message that one gets is the challenge with platform companies is how do you have this platform that can unlock a range of different indications potentially, and but you also have this enormous kind of time lag between the discoveries that you make in the platform and any financial return in the form of a drug. And how do you bridge that gap in a way that gives investors comfort that you can do it while maintaining an active pipeline? And a lot of feedback that I've had from people at other platform companies that I've spoken to over these 4 months has been that in the end, often the platform can churn out more targets than you can possibly hope to develop assets all the way to kind of proof of concept and so on. And that eventually, often these companies face investor pressure to say, okay, you've got these assets that are kind of part way through, you need to focus on getting those all the way through. And then that leads to a deprioritization of the platform. I wouldn't have joined Quotient if I thought that was going to be the way that we were, and because obviously from everything I've said today, I love that discovery science, and I happen to think that Quotient is a great vehicle for continuing to do that discovery science. So I think the challenge that we're wrestling with as a company at the moment is how do we bridge that gap? Do we do it all by partnership? In other words, we just discover the targets and we then hand them off. Do we do some of it in partnership and some of it ourselves? In other words, we take some targets in some diseases through to kind of proof of concept, or do we do all of it by do we kind of just choose our favorite genes and just focus on those and prosecuting them all the way to the end? And I don't think we've bottomed that out. And the other thing that I've learned in these 4 months is that the environment changes on a dime, as it were, and so in the end, the correct answer in 2024 might not be the correct answer in 2025. So I think, and it is one of the beauties of being in startup that you can be quite nimble and you can flex your strategy depending on the kind of prevailing winds that you're exposed to. And so we will, I think, just continue to navigate that. But I would imagine that partnership will be quite a large chunk of the way that we do that. And probably that's the most pragmatic way to maintain a kind of continued investment in the pipeline.
Yeah, and you already have a partnership
Yes.
with Pfizer in both cardiovascular and renal drugs. I'm curious about how that came about and how Quotient is thinking about that partnership now. Obviously, having a big name like Pfizer, obviously you're founded by Flagship in part, but having a big name like Pfizer and a deal is a sign of confidence in your platform. So yeah, curious to hear a little bit more about that partnership and how you're going to make it successful and the potential impact on Quotient.
So this has been a collaboration with Pioneering Medicines at Flagship and Pfizer and Quotient, and as you say, it's hugely exciting for us. This is these are the renal disease and the cardiovascular disease. They're big therapeutic areas in terms of kind of market size and patient populations, and remain kind of areas of unmet need. And they are, frankly, the sort of disease areas where even if we found something and were lucky enough to be able to develop a drug, the idea that we could run a clinical trial in that space and deliver something is just not conceivable. So we really, those are kind of massive disease areas that we have to do in partnership. So for us, it's a really exciting opportunity, and I think what the partnership, what we offer the partnership, if you like, is this kind of unique platform, an ability to discover targets that are otherwise unattainable, but we also get a lot of intellectual stuff coming back from Pfizer in return. So one of the beauties of our platform is that it can be applied to basically any different disease. It also means that we can't be domain-specific experts in all of those diseases. So Pfizer bring a lot of that domain-specific expertise, things around how do we isolate the right cells, what are the right cells in that disease type to be sequencing, how should we think about what any targets in those cell types mean in terms of therapeutic strategies and so on. They bring that domain-specific expertise and can help shape our thinking on this. They often have assays for validation and so on that we can leverage and so on. And then of course they have the might of big pharma in terms of the drug development and testing. So in many ways, I think that ability to bring the kind of different strengths of the two organizations together in this way is what makes this such an exciting opportunity for us.
Yeah, it's interesting that the learnings go both ways in this scenario. And then I guess for our audience less familiar, since heart failure and kidney disease are affect large populations of people, they require very large clinical trials and very hard for a small company to execute on those trials. So it makes sense. And I would say too that it's heartening to see development happening in both cardiovascular and renal areas. I think these areas affect huge numbers of patients, but I would say relative to some other areas like oncology or obesity or some of these others, it's been relatively quiet in cardiovascular and renal therapeutic areas, but they have massive applications and implications for those patients who are affected by those diseases. So it is really exciting to see that your platform has application in an area where there is significant unmet need for new therapeutics. So that's exciting. So you've touched on this a lot throughout this episode around pivoting from academia to startups and your journey all along the way. I'd like to take some time to talk about that process. You were an academic co-founder for Quotient even before you joined on full-time as CSO. So I'm curious on what that was like being an academic co-founder to start, what that was like, and really the mindset shifts and challenges that you faced when you were making that pivot, because they are fundamentally different approaches to science and development, and even with the goals. Obviously some commonalities in terms of innovation and drive to make patients' lives better, but obviously very different type of action-oriented version in a startup, and very broad intellectual knowledge base related topics in academia. So curious on that dynamic and what that was like to move from academia to the startup world.
Yes. So I don't feel I fully transitioned, as it were. To an extent, I feel like I still feel a little bit like an academic in well, probably a sheep in wolf's clothing. um, but yes, it has been an interesting transition. So, you know, the dynamics in academia are quite different in some ways to a startup. So, um, I think one of the really exciting things that I have found in working with Quotient, and I suspect is true of all startups, so small companies, is that is that fundamentally everyone in the company lives or dies by the success of the company, and so this kind of breeds a sense of collaboration and teamwork that I think we've tried to replicate in academia, but it's inherently much harder. Um, and that dynamic has been, it's been hugely fun to work in that kind of team environment, and that sense of everyone having the same shared mission. The other thing that I would say, the other contrast that I would draw between academia and startup land, is that in academia, fundamentally we have a training kind of mandate. We are there to train PhD students, postdocs, technicians, and so on in the craft of doing science. And most of the people doing the science are trainees, and that training mandate requires that those people get trained, that they learn the different aspects of doing science, the process of doing science. That means, a lot of my PhD students and postdocs when I was in academia would do wet lab work and dry lab work. They would kind of master these different trades and different kind of subspecialties within science. And that's, it's great for them. That makes it a very good training opportunity. But it's not, if you're just counting kind of numbers of bases sequenced, or you're just looking at output, it's not necessarily the most efficient way of doing it. And one of the interesting things in startup land is that people wind up, that you have more of a kind of conveyor belt or a kind of production line. So we have people who are experts in kind of flow cytometry. We have people who are experts in laser capture microdissection, in molecular biology, in computational methods, in data analysis, in wet lab experiments. And essentially the projects kind of go through that production line. And it means that people are very kind of, they're working within their kind of expertise and their specialty, and that makes it much more scalable and much more high throughput and probably more efficient. And so the contrast really is that we can industrialize what we were doing in academia to a scale that we struggled to achieve in academia. It's not impossible to do it in academia, and if anywhere does it well, it's Sanger. But still, within the company, we have been able to scale and productionize this to a much greater degree. So those kind of processes, that kind of learning that different way of doing business has been interesting. The other big difference is that in a startup, it seems to me that you're about to run out of money every few months, and so that really focuses one attention on short-term deliverables in a way that in a pressure that I didn't feel so much in academia. One has deadlines and crunches and need to get stuff out in academia, but it's not quite the same sense of impending doom that I think seems to be a permanent fixture of life in a startup.
Yeah, that's a really interesting dynamic that we actually haven't heard before when we talk to founders, is that pressure piece. Obviously less fully exploratory in a way that academia is, where the focus is on, true acquisition of knowledge, as you've highlighted. So that's a very interesting dynamic, and hopefully not too much doom and gloom as you're pursuing these aggressive deadlines. But obviously it focuses the startup's activities to be more action oriented, achieve measurable differences in how the programs are moving forward, obviously for investors, as well as just for the success of the startup to advance a pipeline program or make a concrete development in the platform. So that's an interesting dynamic. One of the other things that's come up a lot from the folks on both the VC and founder side of this about the difference between academia and startups is storytelling. So I'm curious on your thoughts on that in terms of how you communicate in an academic environment, and really what's the difference for how you communicate in a startup environment, which, as you mentioned, you're a little new to, but still curious, as you're making that transition, what comes to mind for you?
So what I would say is that the difference for me is not actually the fact that one is telling stories. One tells stories in both, and actually the stories in both are, they're not a million miles apart in the way that you construct the story. What is different, though, is the mode that you use to tell the story. So in academia, frankly, the kind of mode, the kind of default mode is writing. So we would write our story out. And when I write those stories in academia, I would spend weeks writing paper, getting the paragraphs right, making sure that the kind of tone was right, telling that story in as kind of crisp a way as I could, and I would spend a lot of time crafting that. And that's important because the paper is the permanent scientific record of what I did, and I frankly quite enjoyed that process. In startup land, it's much less permanent. So the storytelling that I do now is in voiceovers, right? So I will have a slide deck and I will be talking to business partners, to investors, to board members, to other people in the company, collaborating companies, et cetera, et cetera. But the story I tell will be a series of slides and it'll be a voiceover. And there's essentially no record of that. Often these are completely confidential. No one, in a year's time, no one will know what I said. And it leads to, it's quite a different sense of storytelling from that point of view. But the actual, what goes into the story, the bits of science, how we stitch those together, the kind of sense of painting a picture of this is what we can do, this is what we have done, this is what we found, this is the eye-opening moment of where you might get to with this. That sense of a journey from idea through execution to potential, that's the same in both, I would say. So that skill set in crafting that narrative journey, I think, is similar in both. It's just the mode of the way that you tell the story is different.
That's a really interesting way of putting it. And I'm curious a little bit here. I found in academia that you want to be more conservative when you're talking about the potential applications. It could cure all the diseases, right? And not to say that you say that kind of framing in a startup, but I think there's a lot more when you're doing like a pitching investors. It's a little bit more sales oriented and saying, I would say, not exaggerating, but taking logical leaps. Hey, if we can identify these mutations, we could have potentially dramatic impacts on a wide variety of diseases. Whereas I don't know if that came up in the conclusion of a academic paper. You might say, like, there's very interesting applications across many diseases. It needs to be studied further. I'm assuming there's some differences when it comes to pitching a little bit more.
There are. I'm going to say that my way of doing this might be quite personal to me. But I would say that some of my papers have been rather ridiculously grandiose. In the end, when we published the liver paper, we had a long battle with the peer reviewers over publishing that paper because they said the discussion that I wrote was way too grandiose. Basically, I was basically saying, like we were talking about with Anastasia earlier about the kind of personalized medicine. I had that in the discussion of the liver paper, but it got taken out because the reviewers hated it. But in general, I think that I would tend to be reasonably grandiose in some of the papers, not all of them, but I would try and paint a picture of where this would go. What I think, coming back to your question about the differences, though, I think I would say I would class the differences more about the story, the audience that you're telling the story to, rather than the kind of level of kind of detail versus potential painting. So if one's writing an academic paper, one is writing for an audience who by and large are pretty expert in the area that you're kind of writing in, and therefore they won't necessarily need some of the kind of high level potential painting that you might need if you were speaking to an audience. So for example, the story that I would tell to an academic audience, if I, about the academic work that I did, when I went and talked to patient groups, which I did quite a lot, or to kind of public, the lay public, it would be the same data. In fact, some of the slides would be the same, but the kind of way that I framed the story and what the potential was would be different for those different audiences. And I feel it's a bit the same in at least in my 4 months thus far in business land. The discussion, the potential, the picture that I would paint for a meeting, let's say with Pfizer, since we were talking about them, around, let's say, a particular cardiovascular disease, if I was meeting with their scientists, this would be right in the nitty gritty of, what can we do? What are the cell types we should do? What disease severity should we be looking at? Should we be looking at untreated or post-treated patients, etc. Right? That picture would be very detailed. But if I was pitching to, some kind of investor who, had good general knowledge in biotech land, but wasn't, a somatic mutation liver and breather like I am, it would be a different level of story that I would be telling, and it would be kind of pitched at the level for the audience to appreciate the opportunity in a way that I wouldn't, necessarily need to in other settings.
That makes sense. So tailoring it to your audience, what their interests are, and the level of detail versus big picture as needed based on the conversation at hand. That makes a lot of sense. So to close out, do you have any final words of wisdom for our entrepreneurs in our audience, maybe who have somewhat similar backgrounds in terms of coming in from academia, who want to make a pivot to starting a company and making a difference for patients? Any final words of wisdom for those folks?
Well, I'm not sure that I'm in a position to give many words of wisdom until this has been more demonstrably successful. But what have I learned? I guess the things that I've learned have been that good ideas require crafting. So I think, and this is one of the things that Flagship does particularly well, is take that initial germ of an idea, like the one that we had and the one that they had, which happened to be the same germ of an idea, but then just inspecting that germ of an idea from every different angle, dressing it down, taking it apart, building it back up again, trying different models, really deeply kind of inspecting it and kind of thrashing out what a business model in that space would look like, where the competition was, being quite hard-nosed about who else might come along and kind of steal your thunder, etc., etc. All of those things were incredibly important for the kind of founding of Quotient. And the more that you can find trusted thought partners to thrash those things out, mentors, we spent a lot of time with mentors at Sanger kind of talking about these ideas. We spent some time talking to other opinion leaders within the space, and kind of the more that you can find people whose judgment you trust and thrashing it out with them and listening to what they say and refining what you're building on the basis of that. I think it's that would be my piece of advice.
Excellent. And on that note, thank you so much for joining the Nucleate Podcast today, Peter.
Thank you. It's been fun.